My name is Michaela TerAvest, and I'm an Associate Professor of Biochemistry and Molecular Biology at Michigan State University. I am so honored to have been selected for a Beckman Young Investigator Award in 2018. Participating in events with the Arnold and Mabel Beckman Foundation has given me a chance to learn more about Dr. Beckman's legacy. Of course, I can't talk about Dr. Beckman without mentioning that we use modern versions of his inventions, the pH meter and the spectrophotometer, every single day in the lab. But what I find more inspiring about Dr. Beckman is his humble nature and practical approach to research and community service. In an interview I listened to about his philanthropic work, Dr. Beckman described how the growth of his company outpaced his financial needs, and that rather than allowing his lifestyle to expand to fit his means, he found a better way to use his assets. Dr. Beckman had the generosity and foresight to see that philanthropy would be a satisfying second career after building his company. Personally, I hope to follow in his footsteps and always look for ways to invest in the future. All of the research in my lab is directed toward ensuring a safer future for humans and for the environment by creating new renewable energy technologies. The BYI Award supports our development of a bacterial strain that will produce biofuels using only carbon dioxide, electricity, and water as the feedstocks. We call this process microbial electrosynthesis. We are very excited about this technology because it could solve multiple different sustainability challenges at the same time. One of the most important aspects of microbial electrosynthesis is that it represents a way to store electricity generated by wind and solar processes in a format that's compatible with existing infrastructure. For example, we know that electricity output from wind farms in Michigan varies over the course of a year. With microbial electrosynthesis, we can even out production by generating fuels when electricity production is high and burning them when electricity production is low. We started this project with a bacterium called Shewanella that already has the ability to accept electrons from an electrode to power chemical reactions inside the cell. One of the things that we're working on is to improve this ability further via genetic modification, including both deleting genes from the Shewanella genome and adding new genes from other organisms. Another bigger challenge is that Shewanella does not have the ability to fix carbon dioxide naturally. To give Shewanella the ability to convert CO2 into fuels, we first added two genes found in photosynthetic organisms. We then made targeted gene deletions in Shewanella to make it dependent on the carbon fixation pathway. We are now using evolution to functionalize the engineered Shewanella strain and hopefully turn it into a voracious CO2 eater. Dr. Beckman's inventions and career have not only accelerated several fields of science, but directly impacted lives around the world. For example, by optimizing oxygen levels in incubators for premature babies and developing a framework to improve air quality in California. I hope that the biotechnologies developed in my lab will eventually also have a positive impact on society, and I thank the Arnold and Mabel Beckman Foundation for helping me launch this project.
Hi, I'm Katie Ford. I'm a fourth-year PhD graduate student working with Dr. Michaela TerAvest in the biochemistry department here at Michigan State University. Broadly, I would describe our research as bioengineering with the ultimate goal of creating an organism that's capable of turning carbon dioxide pollution into useful products like biofuels. We work with a bacteria named Shewanella oneidensis, and the project is focused on genetically modifying this organism to consume carbon dioxide. In order to do that, we had to remove parts of the genome that were considered essential for growth. So what we did was we designed a pipeline by which we can model how this organism grows on a computer and then test different strategies for working around these essential genes in the lab. And we can test these using a CRISPR-based system to confirm the computer's results. And ultimately, this allows us to modify the genome and delete the genes that we previously thought to be essential, which is a really tremendous tool for bioengineering going forward.